Good morning, everyone. Thank you so much for the subscriptions, for the comments, for the shares. I welcome you back. Part 2 of genetics. Yesterday I did introduction on genes. And as I mentioned yesterday, that I'm going to introduce genetics. And in this topic of hereditary science, we are going to know much on DNA, genes, chromosomes. So after introducing genes yesterday, where we stressed much on behind every phenotype there is a genotype which underlies it, and we also saw different scientists with different concepts on how they understood the definition of a gene, and generally hereditary science, how they could define it. But what we saw was it was being crowned in all those their concept, it was being crowned that genetics involves much of the structure, organization, transmission, and functions of those genes, which contribute much to variations. So we generally talked much about the central dogma, starting from the replication process to the transcription to the translation. So at the point the translation takes place, already the gene product, the protein, has been synthesized. So we'll be able to relate the physical expression of a plant, of an organism, of a human, whichever. The characteristics which you can see, the outward expression, the phenotype, you can relate to the genes. So we said the genotype itself has to be there for it to show, for a phenotype to show. So we said the DNA is a component in the gene, and only the DNA is the hereditary material. So today I want us to look at chromosome and its structure before I dive into Mendelian genetics. So by definition, a chromosome, it is more like a thread, but then a self-replicating genetic structure. The most important point there is it contains organized DNA molecule. Because we mentioned yesterday, the DNA will be part of the gene. The gene are hosted by the chromosomes. There are several thousands and hundreds of genes on a chromosome. So that is what we saw yesterday. So the molecule package is found in the nucleus of the cell. We said the nucleus has almost 90% the DNA. And the nucleus, its role is it controls all the activities in the cell. So the chromosome can be seen clearly during metaphase. So if you look at cell division, just as we discussed in the year 1, cell division had the interphase, and then now you are entering into the mitotic phase, where we had the anaphase, we had the metaphase, we had the telophase, the prophase. So those were the mitotic phase. But in the interphase, which is known as the longest phase of the cell cycle, you don't find the metaphase. So the metaphase is found on the mitotic phase, and where we could always explain that now you could see there is an equatorial plate forming. So you'll always see them aligned at the center. So the chromosomes can be clearly seen during metaphase when you stain them with the correct dye, of course, and then you view them under light microscope. That is when you can clearly see the chromosomes. So Strasberg in 1875 is the one who discovered the thread-like structures of the chromosome, which appear during cell division. So based on whatever the findings Strasberg did, you already saw that the thread-like structures possibly could be chromosomes. But then in all types of organisms, be it the eukaryotes, be it the prokaryotes, but then in eukaryotes and the higher organisms, the chromosomes are well organized. So they are well organized such that the nucleus will contain a definite number of the chromosomes, the definite size, as well as shape. So we talk about morphology when we look at how organized the chromosomes are in those high organisms. While they are also still try to understand much on the chromosome, definition of the chromosome or the term chromosome by 1888, and then it will help much in what Strasberg has already proposed. So it helps much to understand the structure of the chromosome as well as the function. So we have somatic chromosome number and gametic chromosome number. When you talk about the somatic chromosome number, it refers to the diploid state, the 2n. So the number of chromosomes found in somatic cell will be represented as 2n in diploid. And then we have the gametic chromosome number where it's half the somatic. It's half 2n. So you talk about the haploid. So you talk about the gametic chromosome number being haploid as well as the somatic chromosome number being diploid or 2n. Sometimes you'll always mention homologous chromosomes. So the two copies of chromosomes which are identical in morphology, gene content, as well as gene order, then we'll refer to them as homologous chromosome. And most of the cases where you talk much about homology, you're talking about they have the same ancestral origin, but maybe at some point by speciation the organisms were differing based on speciation or based on duplication, then it has to bring about a copy of its own. So we talk about mutation. The other one, we talk about speciation, but then they have a common ancestry. So the homologous chromosomes we refer to as the two copies of chromosomes, but then they are identical in morphology, gene content, and gene order. We'll be able to see an example of the same when we look at the family, for example, of Homidae. You talk about the Homo sapien, you talk about the apes. So as much as we are from the same ancestor, common ancestry, but due to speciation, there is a gene which is found on man, a gene found in gorilla, a gene found in orangutan, because we are of the same family. So we talk about the same ancestral. We have the common ancestor. So we are homologous. We have a common ancestor. But due to speciation, the same gene which is found in man and found in gorillas, it has the same function, but it could be found on a different chromosome. So it tells much when you do BLAST, still in bioinformatics. BLAST is what you're trying to look at, the common ancestry of different DNA sequences of those species which you're trying to look at. You're trying to find a root. You're trying to draw a phylogenetic tree, how related the organisms are. So we'll be closer to the apes than dogs. There is a gene which makes us to come closer together. So if you do BLAST, we'll find close relationship. You'll find high hits. Based on the hits, you'll get we are close to the apes than maybe the cat's family or the dog family. But still, there is still also a gene in the dogs and the cat family which can also try to trace that we are from the same common ancestry. So that is the homologous chromosome, two copies of chromosome, but ordinarily identical in morphology, gene content, and gene order. There are two types of chromosomes. We have the autosomes and then the sex chromosomes. So we talk about the autosomes. The other ones who do not carry the sex, they do not determine the sex. So we have the X and Y's, especially when you talk about the sex chromosomes in human. So the male will have always the XY, the female will have the XX, and at that point we talk about the sex chromosomes. So the autosomes, they are the ones which control other characters other than the sex characters. So autosomes will only control a phenotype, a trait. So we talk about they carry genes for somatic characters. Then we have the sex chromosomes, which are involved in the sex determination. Just as I've mentioned, in human we'll have the XY for the males, and the females will have the XX. The sex chromosomes, they are also known as the gonosomes. So humans and most other animals, or most other mammals, they have 2 sex chromosomes, X and Y. Also known as heterosome. It is an heterosome when it appears as XY. So it is heterogametic. But when it is XX, like females, you talk about homogametic. So we have the XYs as the sex chromosomes. So the females have 2 chromosomes in diploid cells, and we talk about the XX. The males have the X and a Y chromosome, meaning the XY. But in birds, it's different. It's the females who are heterogametic, the ZW, and then the males are homogametic, ZZ. But remember, in humans, the females are XX, so they are homogametic. But now, in birds, the female are heterogametic, we have the ZW, while the males now in birds are homogametic, they are ZZ. The males in human are heterogametic, they are XY. So those are sex chromosomes, also known as the gonosomes. So we already talked about the diploid and the haploid, the somatic and the gametic. So in this case, when you talk about the diploid cell, we have the 2N, where N will always represent the chromosome number. So it will have the 2 homologous copies of each of the chromosomes. Then the haploid will have half the copy, only 1 copy of each of the chromosomes. So in animals, the gametes are always sperms and eggs, for example, and they come in haploid state. So you'll have the male contributing a gamete, the female contributing a gamete, but then we have the sperms have to contribute from the side of the male, as well as the eggs from the side of the female. So the chromosome number will always vary as per any of those organisms. So we are still talking about the somatic and the gametic. So in somatic state, you talk about the diploid number, the 2N, and then the gametic, we are talking much about the haploid, or the half 2N. So the number of chromosomes will always vary from each of those species to species, and normally all individuals of all species have the same chromosome number. So I was still talking much about the family of the Homidae. If you look at their chromosome number of man, of gorilla and orangutan, as much as in diploid state we have 46, you'll see orangutan has 48, gorilla has 48. Just a slight difference in the chromosome number. But if you look at the gene order morphology, it's similar. So it varies from species to species. But if you find that even from the cat family, you look at the chromosome number of the cheetahs, of the lion, for example, it's more or less the same. So the dog family, the cat family, you look at it, there is a similarity. It talks much about the homologous chromosomes. It tells much about the common ancestry. But at some point, speciation takes place, and each of those genes are in those chromosomes, but then different chromosome number. It could be a different chromosome on those species, but due to speciation, they were separated, but they have a common ancestry. So normally all individuals of a species have the same chromosome number. So the size of chromosome, we just saw that we can clearly see it during metaphase, and then they talk much about its size being short in fungi and some birds, and being long in some plants. So it also varies, the size will always vary. So each chromosome will always have the short and the long arm, and then we have a primary constriction which separates the 2 arms. So you talk about the centromere. But now if you want to know that P is the shorter arm from the French word petite, which is small. I don't know how the Q came about. I think Q follows P. So P became the short arm, the French word for petite, and then the Q is the longer arm. So we have the P, the shorter arm, and the Q as the longer arm. So it tells much of the shapes of those chromosomes based on the primary constriction. There is a primary constriction in between the two arms. It separates the two arms, so it is the one which is known as the centromere, and it determines much the chromosome shape. So the centromere, or the primary constriction, makes those chromosomes to be classified as the subterminal, the ones which can be found towards the middle. So you talk about the centromere being placed above the middle, or being at the middle, or being at the tip. So we are going to look at different classification of chromosomes based on the position of the centromere or the primary constriction. So you talk about the homologous chromosomes, which we mentioned in the previous slide. We are trying to look much that they look the same in the gene morphology, shape, and order. But then the main idea here is, for the homologous chromosomes, we look at there is a common ancestry. There is a relationship between these species, however much the chromosome number differ species per species. But if you look at their common ancestry, they have the same, the common ancestor. So diploid organisms will always have two copies. And you talk about the two copies, it was the somatic one where we talk about the 2N. So both of the copies are identical in morphology, gene content, gene order, and at that point is when they are referred to as homologous chromosome. So what is also important about those homologous chromosomes is they are inherited from the parents. So one homolog comes from the father, and then the other one comes from the mother. So they have to contribute. Each one of them has to contribute these chromosomes. And then at the point of segregation, recombination will take place. And at that point is when the characteristics will be passed down to the offsprings from their parents. It talks much about the Mendelian principle of segregation. So chromosomes are classified based on the constriction, the primary constriction, the position of the centromere. So based on the position of the centromere, the chromosomes are classified as acrocentric, submetacentric, metacentric, and telocentric. The last one is telocentric. So we are going to look at each one of them: the acrocentric, the metacentric, the submetacentric, and lastly the telocentric. So we are still talking about the P being the short arm, petite, just as you see there, small, it means the French word for small. Then the Q will automatically go for the long arm. So we are looking at the metacentric chromosome. Classification of those chromosomes, or looking at the types of chromosome based on the position of the centromere, the primary constriction point. In this case, if you see the meta, you look at it, it will be placed. The centromere will be placed in the middle, such that the long arm and the short arm is 50-50 divided. So they'll always see them in a shape of a V shape during anaphase. So the centromere is located at the center, not in the center of the chromosome. So we talk about the median. And then we talk much about the shape during anaphase. At anaphase, you'll see a V shape. Anaphase also still falls under the mitotic phase. So the metacentric chromosome take a V shape during anaphase. You can draw it this way, you can draw it this way, but what you have to know is both arms are equal. It is 50-50. We are classifying them based on the primary constriction, the position of the centromere. Submetacentric, it is a little bit up from the middle point. So it's located on one side of the central point of a chromosome. So it's like one end is, it is like 3/4. One end is like 40%, the other one is 60. It's not now 50-50, but there is a percentage which has gone much to one side. So it is above, a little bit, slightly above the middle point. So the centromere is submedium, giving one longer and one shorter. So they always have an L shape during anaphase or a J shape. So you see how we write a J? There is somewhere which is a bit smaller portion than the other one. So it is very important to mention the position of the centromere when you talk about submetacentric or when you talk about metacentric, and also it's important to mention the shape during anaphase. Acrocentric, it is towards the end. So the ends are always known as the telomeres. It is towards the end, but not. So we can talk much about 20% and 80. The long and the shorter, 20 to 80%, a ratio of 20 to 80. So the centromere is located close to one end. The centromere is close to one end. It is more terminally placed and forms very unequal arm length. So the name refers to acro, towards the peak, towards the peak. So the P arm is so short that it is difficult to observe, but it is still present. So it gives a rod shape. During anaphase, you'll see it as a rod shape. Then we have the last bit called telocentric. The telomeres, they're the ones which give stability to the chromosomes. So the ends. So if you see at this centromere, you can't even see the shorter arm because the centromere is at the end, is at the telomeres. So it is located at the end of the chromosome. At the terminal, it lies at one end. So it will bring a rod-shaped chromosome during anaphase. So it shows much the position of the centromere is at the ends, at the telomeres. So we know it's known as telocentric chromosome. Then there is also classification based on the number of the centromeres. It can be acentric, the one without the centromere, monocentric, the one with one centromere, dicentric, the one with two centromeres, and polycentric, the one with more than two centromeres. So we had to introduce chromosomes so that when we dive into Mendelian genetics, we'll be able to understand much what genes are, what chromosomes are, and we'll be able to understand the hereditary